When is stress good for you? How stress works in the human body to make or break us. This is an Aeon Essays, AEON. The subtle flows and toxic hits of stress get under the skin, making or breaking the body and brain over a lifetime. This article is by Bruce McEwen. It was an Alfred E. Mirsky Professor of Neurosciences and Behavior and head of the Harold and Margaret Millikan Hatch Laboratory of Neuroendocrinology at Rockefeller University in New York City. His award-winning research on stress and the brain has been published in Proceedings to the National Academy of Sciences, the Journal of Neuroscience, and Molecular Psychiatry. He lives in New York. This is edited by Pam Weintraub. Stress pervades our lives. We become anxious when we hear of violence, chaos, or discord. And in our relatively secure world, the pace of life and its demands often lead us to feel that there is too much to do in too little time. This disrupts our natural biological rhythms and encourages unhealthy behaviors such as eating too much of the wrong things, neglecting exercise, and missing out on sleep. Racial and ethnic discrimination, along with lack of educational opportunities and economic advancement, take their toll on a large segment of the population in the United States. Incarceration is the rule rather than the exception for some of the most vulnerable. Adverse experiences in infancy and childhood, including poverty, leave a lifelong imprint on the brain and body and undermine long-term health, increasing the incidence of cardiovascular disease, diabetes, depression, substance abuse, antisocial behavior, and dementia. How does all of this stress get under our skin? What does it do to our brains and our bodies? What can we do about it? And is stress so multifaceted and pervasive that we could have trouble controlling it at all? The psychologist Jerome Kagan at Harvard University recently complained that the word stress has been used in so many ways as to be almost meaningless. He suggests it's warranted only for the most extreme circumstances and damaging events. But my decades of experience suggest another approach. The insidious power of stress to get under the skin was the focus of a MacArthur Foundation research network that I joined more than 2 decades ago, uniting me with social scientists, physicians, and epidemiologists around a common problem: how to measure and evaluate stress from our social and physical environments. Our collaboration continued under the auspices of the National Scientific Council on the Developing Child, has shown that stress acts on the body and brain, profoundly influencing health and disease. Our findings are nuanced, starting with the fact that not all stress is the same. Good stress involves taking a chance on something one wants, like interviewing for a job or school, or giving a talk before strangers and feeling rewarded when successful. Tolerable stress means that something bad happens, like losing a job or a loved one, but we have the personal resources and support systems to weather the storm. Toxic stress is what Kagan refers to, something so bad that we don't have the personal resources or support systems to navigate it. Something that could plunge us into mental or physical ill health and throw us for a loop. Now let us put these 3 forms of stress into a biological and behavioral context by invoking homeostasis, the physiological state maintained by the body to keep us alive. It is through homeostasis that we maintain body temperature and pH, alkalinity and acidity within a narrow range, keep our tissues perfused with oxygen and our cells fed. To maintain this steady state, our body secretes hormones such as adrenaline. Indeed, when we encounter an acute perceived threat, a large menacing dog, for example, The hypothalamus at the base of our brain sets off an alarm system in our body, sending chemical signals to the pituitary gland. The pituitary, in return, releases ACTH, adrenocorticotropic hormone, that activates our adrenal glands next to our kidneys to release adrenaline and the primary stress hormone, cortisol. Adrenaline increases heart rate, blood pressure, and energy supplies. Cortisol increases glucose in the bloodstream and has many beneficial effects on the immune system and brain, among other organs. In a fight or flight situation, cortisol moderates immune system responses and suppresses the digestive system, the reproductive system, and the growth processes, as well as signaling brain regions that control cognitive function, mood, motivation, and fear. Biochemical mediators such as cortisol and adrenaline help us to adapt as long as they are turned on in a balanced way when we need them, and then turned off again when the challenge is over. When that does not happen, these hormones of stress can cause unhealthy changes in the brain and body, for example, high or low blood pressure or an accumulation of body fat. When wear and tear on the body results from imbalance of the mediators, we use the term allostatic load. When wear and tear is strongest, we call it allostatic overload. And this is what occurs in toxic stress. An example is when bad health behaviors such as smoking, drinking, and loneliness result in hypertension and belly fat, causing coronary artery blockage. In short, the mediators that help us to adapt and maintain our homeostasis to survive can also continue to the well-known diseases of modern life. The word stress is often explained as a fight or flight response. But what really affects our health and our well-being are the more subtle, gradual, and long-term influences from our social and physical environment, our family and neighborhood, noisy and polluted environment, being lonely, not getting enough physical activity, eating too much of the wrong foods, smoking, and drinking too much alcohol. All these contribute to allostatic load and overload through the same biological mediators that help us to adapt and stay alive. Even though we know all this, we often hear that measuring our cortisol levels will tell us if we are stressed. This reflects a misunderstanding at two levels. First, a single measure of cortisol will tell us nothing, since cortisol levels go up and down without minutes. And this halting this fluctuation impairs ongoing adaptive plasticity within the brain. Moreover, cortisol fluctuates throughout the day, going up in the morning to awaken us and then declining, except for a rise at lunchtime until it falls to low levels in the evening before we go to bed. Flattening this diurnal rhythm is a consequence of sleep deprivation and certain forms of major depression. A flat rhythm not only attenuates a robust adaptive cortisol stress response, but it also promotes obesity and high cholesterol. Risk factors for diabetes and cardiovascular disease. It does so in part by causing the liver to make the ingredients to deposit body fat. There are several ways to measure cortisol to determine whether the normal daily rhythm has derailed. We can collect urine overnight or over the day. We can measure cortisol in hair from the forehead, which gives us an index of our cortisol production over days, or we can measure cortisol in our saliva multiple times during the day. During and after a stressful challenge, such as talking about something personal before a group of strangers. The stressful challenge gives us a picture of the efficiency of our allostasis, marked by turning up our cortisol response when challenged and needed for adaptation to maintain homeostasis, and then turning it off when the stressor is over so as not to produce adverse effects of allostatic load and overload. Failure to turn on cortisol when needed is bad, leaving the door open for the body's inflammatory response to compensate in an imperfect way. Too much inflammation can kill us in septic shock. Failure to turn off cortisol after the stress is over produces negative effects too. upon the consequences of an increase in fat production, leading to obesity, diabetes, depression, and eventual heart disease, all contributors to allostatic load. Given our need for a robust cortisol response in the face of stress, the second misunderstanding about cortisol is the notion that it's a bad guy. Rather, cortisol has a normal physiological role. It helps us adapt to stressors and coordinates our metabolism with daily activity and sleep patterns. We would not live very long or well without cortisol. As my former student, Firdous Dabar, now a neuroimmunologist at the University of Miami, found, the early morning rise in cortisol, along with the stress response, activates immune function so that we can fight an infection or repair a wound. Likewise, the normal morning awakening rise of cortisol that helps rouse us and make us hungry for breakfast enhances the body's response to immunization if administered in the morning. The body's response is like an orchestra involving many players working in harmony. If the body functions like an orchestra, the conductor is in the brain. It stores memories from bad as well as good experiences and works with the body to keep us alive by minimizing those subtle and long-term influences that cause allostatic load and overload. What we call the wisdom of the body refers back to allostasis, the active process of biological adaptation and its role in maintaining homeostasis. Indeed, the brain is a plastic and vulnerable organ, continually sculpted by experience. It changes its architecture and function as part of the allostasis. One recent study showed how the brain architecture of a mother is sculpted during pregnancy as part of the formation of attachment to the child. Other studies show how musicians' brains develop with greater skill, leading to an increased neuron size and increased connections between sensory and motor control regions of the cerebral cortex. As opposed to motherhood and musicianship, toxic stress can increase anxiety by causing neurons in the amygdala, a brain region controlling anxiety and aggression, to become larger. Mindfulness practices such as meditation can reverse the process and reduce the size of those neurons, along with the stress. And physical activity such as walking every day causes genesis of new neurons in the hippocampus, a brain region that is essential for daily memory and spatial orientation. And it also improves memory and mood. We also need to consider where our genes fit in and understand that they do not rigidly determine our destiny, but rather provide the foundation on which our experiences shape our brains and bodies over the life course via epigenetic mechanisms, which operate above the genome, controlling the expression of genes without changing the genetic code. Epigenetics drives the seamless integration of experiences, both good and bad, acting on our genetic code over our life course. We now understand that epigenetics is a means by which stress acts on the body, the genome, and the brain. My life's work has helped me to tell the story of stress, starting with the mentors for my dissertation completed in 1964. These two Rockefeller University scientists, Vincent Alfrey and Alfred Mirsky, taught me the fundamentals of epigenetics in the 1960s, before there was much interest in it. And when epigenetics meant something quite different, namely, the emergence of characteristics of a fertilized egg developed in a living organism. Development from embryo to independence is programmed into each species, but the individual characteristics that emerge are influenced by experience. And that is where the modern use of epigenetics comes from. An example of this is a pair of identical twins with genes that predispose them to schizophrenia or bipolar illness. Even with the same DNA, the probability that one twin will develop the disease when the other twin gets it is only in the range of 30 to 60%, which leaves plenty of room for experiences and other environmental factors to either prevent or precipitate the disorder. Alfrey and Mirsky studied proteins called histones, which package and order DNA. Histones can be chemically modified to unwind the double helix. allowing genes to be expressed. Around 1960, researchers showed that hormones such as cortisol and estradiol use this mechanism to turn on genes in the uterus and liver, and this became the focus of my work in 1966. Before long, I changed my focus from the liver to the brain. As with cortisol in the liver, hormones of the adrenals and gonads could alter gene expression in the brain, working synergistically with other biochemical mediators to alter brain structure and function. Because experience itself affected these hormones, experience molded what was now called the epigenetic effects. This led to the finding that the adrenal stress hormone cortisol acts epigenetically on a brain structure called the hippocampus, which we now know mediates memory of daily events in space and time and also regulates mood. In other words, the hippocampus is the GPS of the brain, a discovery that in 2014 saw the Nobel Prize awarded jointly to the UK-based neuroscientist John O'Keefe and the Norwegian scientists May-Britt Moser and Edvard Moser. The hippocampus has since become a gateway into learning how sex hormones, metabolic hormones, and stress hormones enter the brain, bind to receptors, and act epigenetically to positively regulate structure and affect our behavior. It has also helped us study conditions of toxic stress, when the same hormones and mediators contribute to allostatic overload, and when that happens, the organs of the body, including the heart and brain, are targeted for damage in a toxic storm. Over the course of decades, my laboratory participated in and in some cases initiated these discoveries with the help of some remarkable students, postdoctoral fellows, and colleagues. Among them is Ron Duclos, now a professor at the University of Leiden, who studied the impact of synthetic glucocorticoids, which serve as potent inhibitors of inflammation and immune function, and stimulators of liver glucose metabolism, hence the name glucocorticoid. Cortisol is a natural glucocorticoid, and Duclos found that synthetic glucocorticoids, such as the medication dexamethasone, DEX, are actually excluded from the brain while cortisol gets in. But when a medication is given to quell inflammation, it can shut off the body's ability to make cortisol. Then, when DEX treatment is terminated, the body and brain become deficient in cortisol, causing terrible mood swings and metabolic and immune disruption. After that, Duclos went on to show in his own laboratory with his studies Hans Reil, now professor at the University of Bristol, that cortisol in the hippocampus binds to two receptor types called MR and GR to produce its myriad of important actions in the brain. And another important advance was made by a student in my laboratory, Robert Sapolsky, now a professor at Stanford, and a well-known author of a number of books, who found that over the lifespan of a rat, the cortisol equivalent in the rat corticosterone gradually causes wear and tear on the hippocampus, impairing not only memory and mood, but also the ability to shut off the production of its glucocorticoids. This effect is more evident in animals and people who have experienced toxic stress. The glucocorticoid cascade hypothesis of stress and aging, as it is called, was the basis for the concept of allostatic load and overload. Sapolsky also did seminal work on dominant and subordinate baboons in Africa, and laid the groundwork for how income, education, and human social interactions impact physical and mental health. Until we grasped the impact of epigenetics, the brain was regarded as structurally stable in adult life, and the main focus for understanding normal and abnormal brain function was neurochemistry and neuropharmacology. During the 1980s, practitioners relied largely on antidepressants such as Prozac, along with an array of antipsychotic medications to help patients heal. Then in 1988, Elizabeth Gould, now a neuroscientist and professor at Princeton, came to my laboratory as a postdoctoral fellow. She introduced us to an old method hailing from the late 1800s and to Camillo Golgi, an Italian neuroanatomist who won a Nobel Prize for it. The Golgi technique, when done right, allows the investigator to visualize and measure the dendrites, like tree branches emerging from neurons, and even the spines, sites of synapses or connection with other neurons on those dendrites. Using the Golgi technique, Gould could together with the Japanese biological psychiatrist Yoshifumi Watanabe, showed that dendrites shrink and spine synapses are lost on hippocampal neurons after chronic stress lasting several weeks. The effect was due in part to the action of glucocorticoids such as cortisol. In contrast, Kathleen Woolley showed that spine synapses come and go during the rat's estrus cycle compared to the human menstrual cycle, because of the fluctuations of the ovarian hormones estradiol and progesterone. Remarkably, in both cases, the hormones did not work alone and required, among other mediators, the main transmitter in the brain, glutamate. Thus, circulating hormones not only enter the brain and bind to receptors, but also participate in the brain's own neurotransmitters in what we call adaptive plasticity. Structural changes in the brain to enhance our success and survival. Adaptive plasticity underlines behavioral neurological adaptation to the world. For example, shrinkage of dendrites in the hippocampus protects those neurons from damage by overstimulation during toxic stress. Cyclic fluctuations of spine synapses during the estrus and human menstrual cycle underlie differences in behavior, including mood swings. The actions of estradiol on cognitive function and their absence after menopause have become the focus of hormone therapy to slow cognitive aging and prevent Alzheimer's disease, and the work on this topic by my colleague John Morrison, a director of the Primate Research Center at University of California, Davis, some in collaboration with us, have been very influential. Likewise, the contributions by our former postdoctoral fellow, Roberta Brinton, now professor at the University of Arizona, have opened new avenues for use of the hormone progesterone as a protective agent for the aging and damaged brain. Gould and her students, Woolley and Heather Cameron, now principal investigators at the U.S. National Institute of Mental Health, also established that neurons of the dendritic gyrus, part of the hippocampus, die and are replaced via the process of neurogenesis, which continues over the entire life course. They found that toxic stress suppresses that neurogenesis and shrinks the hippocampus, while other laboratories went on to show that physical activity increases neurogenesis, not only in young, but also in older animals. These revelations about adult brain neurogenesis have huge implications, not only because of the recognition that stem or progenitor cells might be used to treat brain damage, but also because of their meaning for lifestyle. Regular physical exercise increases this neurogenesis in old as well as young people, and improves memory and mood, and even enlarges the hippocampus, which tends to shrink in depression and diabetes, among other conditions. Within 6 months to a year, regular aerobic activities such as walking an hour a day, 5 out of 7 days a week, not only makes the hippocampus larger and improves memory, but it also improves decision-making by improving blood flow and metabolic function in the prefrontal cortex, a brain region essential for self-regulation of emotions and impulses, as well as working memory. Indeed, regular physical activity is the most important behavior that one can do to maintain brain and body health. And as a further illustration of brain-body communication, the ability of exercise to stimulate neurogenesis requires that at least 2 hormones be taken up from the body into the brain. One of them, IGF-1, comes from the liver, and the other, capsaicin B, comes from muscle. Plasticity of the brain extends to the diurnal cycle of waking and sleeping, and reaches beyond the hippocampus to other brain regions. A former student, Connor Liston, now an assistant professor of psychiatry at the Weill Cornell Medical School, found that some but not all synapses in many parts of the cerebral cortex turn over during the day-night cycle due to the fluctuation of cortisol. Interfering with that cycle at the wrong time of the day interferes with motor learning, example, learning to play golf. Considering how many ways we modern humans interfere with our natural day-night rhythm, for example, by turning on a light in the middle of the night, this is a lesson to all of us to give the wisdom of the body a better chance to help us. Another way that we interfere with the natural cycle is the shift work and jet lag. Our former postdoctoral fellow, Ilya Karastaris, now an associate professor at Washington State University, found that creating an animal model of shift work caused dendrites in the prefrontal cortex, the brain region that governs our ability to regulate emotions and impulses as well as working memory, to shrink in the animal to become cognitively rigid when challenged with a memory task that required changing the rules. Moreover, the shift work animals became fatter and insulin resistant, signs of prediabetes and depressive-like behavior. Shift work in our own species is associated with greater obesity, diabetes, cardiovascular disease, and mental health problems. The prefrontal cortex also responds to what we call tolerable stress. During his MD PhD thesis research, Liston assessed a group of medical students for perceived stress, how much or little they felt in the control of their lives. He found that those with the highest perceived stress were slower in doing a cognitive flexibility test and also had slower functional connectivity in a brain circuit involving the prefrontal cortex when tested in an MRI machine. The reason we can call this tolerable stress is that after a vacation, these impairments disappeared, showing the resilience of the young adult brain. Parallel studies of perceived stress on an animal model allowed Liston to see shrinkage of neuronal dendrites and reduction of synapses in the prefrontal cortex that explain the deficits in cognitive flexibility. To complete the story of brain plasticity, we need to describe how the same stressors cause dendrites to shrink and synapses to be lost in the prefrontal cortex and hippocampus. The answer comes from Sumantra Chatterjee, a professor at India's National Center for Biological Sciences in Bangalore, and his team. Dendrites in the basolateral amygdala, the seat of fear and anxiety and strong emotions, grow and become more branched, increasing a sensation of anxiety. Liston found that dendrites in the orbitofrontal part of the prefrontal cortex also expand, increasing vigilance. In the short term, these changes may be adaptive because anxiety and vigilance can aid us during dangerous and uncertain times. But if the threat passes and the behavioral state gets stuck and persists along with changes in neural circuitry, such maladaptation requires intervention to open windows of plasticity with a combination of pharmacological and behavioral therapies.